All right, welcome everyone to another StaPH-B call. Today we're going to continue on the focus on SARS-CoV-2 sequencing, and then this time we're also focusing on the aspect of genomic epidemiology. So we invited Ellsworth Campbell from the CDC to speak with us today about MicrobeTrace. This is a platform that was not originally developed for SARS-CoV-2. They have years of development for other different infectious disease investigations. But I think through Ells's presentation, you're going to see how it can bring a lot of utility to epidemiological investigations regarding COVID-19. And then also, Els is going to give us some information for the epidemiologists that we've invited on this call too, to how they can start integrating these sequencing efforts that a lot of labs have been focusing on to help inform their public health decision making. So with that, Els, I'm going to go ahead and mute myself, and you can take over.
All righty. So my name is Ellsworth Campbell. I'm a computational biologist in the Division of HIV/AIDS Prevention at CDC, and I'm the project manager for a program called MicrobeTrace. And I'm going to describe how we built it, what its use case is, but really try and hone in on how we can use it for SARS-CoV-2. And also, I just want to make it clear, I love being interrupted, so if there are questions, please feel free, whether that's during the slides part or during the demo part. So I'm going to start with some background. Some of you may be aware that the president announced the ending the HIV epidemic initiative about a year ago. And this is really kind of a moonshot effort to try and reduce HIV transmission by 90% over 10 years. Now, there are 4 pillars to this response strategy, and diagnose, treat, and prevent are all relatively separate approaches to the problem. However, response, really identifying where we need to do these other 3 pillars, factors into all of them. Okay, and so what we're going to focus on is how we use MicrobeTrace for HIV response, and hopefully you'll be able to make some analogies to how we can use this in other ways. So I'm going to take a step back and say, well, why build transmission networks? Whether this is contact tracing networks, genetic distance networks, it doesn't matter in particular what you're using to connect people. The concept here is that the person A had contact with person B is directly related to the concept of HIV virus A or virus B are this distant from one another. So these 2 types of data structures overlay on top of each other very nicely. And so again, this is for HIV, but we've used this for TB, we've used this for SARS-CoV-2, for a variety of other pathogens. The concepts hold true across the board. And doing this integration of the human perspective of transmission versus what the genetic sequences tell us allows us to inform fine-grained, resolution of what the transmission dynamics look like that allow us to look at outbreak-specific interventions for this outbreak, as well as develop future prevention strategies. They also allow us to identify links between cases when people don't tell us anything. So we can look at sequences and say, hey, these 2 sequences are identical. They're very, very similar to one another, but we don't have any behavioral link between the 2. Let's try and fill in the gap here by stitching these 2 bits of information together. So let me show you what that looks like. We can start with what the epidemiologic network looks like. For me, that might be a high-risk sexual network, but in the context of COVID, that just might be contacts for a brief period of time. We integrate that with what the lab data say, or what the genetic information say. And this is really comparing the sequence data that we have available, and then drawing lines between those sequences when they're very closely related to one another. And then when we look at those 2 integrated data sets, we can see where they agree and where they might disagree. But in this situation, when we integrate both of them, there's a lot more lines here on screen than there are actual transmission events that could have occurred to generate this cluster. So we can apply informatic tools like minimum spanning trees to try and hone in on which ones are the most likely links that connect these individuals. Then we can layer on additional information to come up with really, what are we going to do? How would we respond to this? For example, here we'll layer on the recency of HIV infection to get an idea of where that cluster is growing most rapidly. So for this network, often we face the question of, when I get a network, what do I do with it? How does it translate to public health action? Well, for us, up in this top left corner where there seem to be 2 very acute recent infections, we might want to do partner services follow-up to identify their partners and kind of find the leading edge of transmission. But this person that's really, really centrally connected at the bottom, we might want to follow up with antiretroviral therapy to potentially reduce transmissibility, right? So we can use different aspects of this network to try and identify different prevention strategies that make sense. So in the real world, how do we put these to use? In 2015, there was a large HIV outbreak among persons who inject drugs, primarily opioid driven. And basically, we were able to integrate the contact tracing data and the genetic information from comparing their sequences to come up with a picture of what that outbreak looked like. And this really allowed us to say, hey, if you look at this cluster, there were multiple bottlenecks here. There were points of intervention where if we had intervened, say, at this position, we might have been able to cut off all these downstream infections. So for this particular outbreak, when we started, we collected the contact tracing data. And what you see here is a tangled mess, right? Each circle represents a person. Each line represents a connection or a type of high-risk contact between those individuals. The circles are sized by the number of high-risk contacts that each person reports. And then they're colored by HIV infection status. So in this outbreak, there were a lot of injection drug use. People were injecting 4 to 15 times a day with 1 to 6 partners per injection event. And there was quite a lot of hepatitis C co-infection. So what do you do with these data? Like, how is this useful? This is a tangogram. Like, how do we turn this into public health? Well, what we can do is count up the number of types of lines for each individual with respect to the likelihood of them being positive or negative in that diagram. So we applied a machine learning algorithm to the network that's collected from contact tracing. And basically what we were able to do is find split points in the data, find things that were very predictive of having an HIV positive status. So in this bottom right corner of this decision tree, you can see that individuals with 4 or more injection drug use contacts were HIV positive 93% of the time. If you had 3 or less, then you were only positive 16.8% of the time. Now, among those, you can further divide those up and kind of find your higher and low risk people. None of this involves sequence data. So this is how you can turn this kind of a tangogram diagram into actionable intelligence for public health. So enter MicrobeTrace. That's kind of like a high level background on how we use these types of diagrams, what they're useful for in epidemiology, and how we can integrate them with lab data. MicrobeTrace really was built after that particular outbreak investigation that I described to enable that type of an investigation, to make it easier to spin up on a moment's notice. So MicrobeTrace enables network analysis, both of the sequence data and of the contact tracing data, and of the integrated view of both of those data sets. And it's pathogen agnostic. It doesn't matter what sequence set you're using. We've worked with quite a lot of different pathogens. This is an old slide, so there's a lot more than what we have so far. Ultimately, if you've got a FASTA file of a consensus sequence, if you've got pairwise genetic distances in a matrix, we can work with you and visualize those data. Any questions up to now? All right.
So one quick question is, what is the viral evolution foundation in order to build up this big network? And I do remember for HIV transmission, the threshold is less than 2% of the whole genome, the HIV whole genome, right?
Right. So the nice thing is MicrobeTrace really supports you making whatever call you would like to make in terms of the viral evolution. So we, in HIV, we focus on the polymerase region, and most of that is based primarily on it's an available sample. It's sequenced for every individual that's diagnosed with HIV, and so that's why it's widely available. It may not be the best for reconstructing transmission, but it is widely available. And so it's a relatively established method to measure pairwise genetic distance, and if they're close beyond a certain threshold, we typically use 1.5%. Then we draw a line between them. Ultimately, MicrobeTrace is designed to support your investigation. So if you've already built a Newick tree, if you've already computed your pairwise genetic distance by whatever method you deem fit, we can support inputting of those types of files. So if you look here, this is what the MicrobeTrace interface looks like. You navigate to it via a web browser. Okay, you go there with Google Chrome or another modern web browser. As soon as you navigate to the page, it downloads the entire program to your computer, so that from that point forward, you can disconnect from the internet. It no longer needs the internet. This program functions in an entirely headless or client-side way. And so in that sense, we both will compare FASTA sequences according to the tRNA distance model. We'll compare SNP arrays or SNP comparisons at a Hamming distance model. These are options that we provide to you kind of in the advanced features. So there's a lot of customizations that allow us to say we're not really making too many strong assumptions for viral, bacterial, or any kind of evolution. We let you come with custom trees if you'd like. So this, what I'm showing here is a slightly outdated screenshot. We've since added Newick files, which you'll see when I do the demo in a moment. Does that answer your question?
Yes.
So what types of visuals can we make? What I'm showing here are contact tracing data that were collected by the Korean CDC in response to COVID-19 relatively recently. These are publicly available. And so each line represents a type of contact, and each person represents an individual. You can see that there's a little number over each node, and that is the number of high-risk contact reports for each person. We've also got an incidence curve based on date of symptom onset. We've got maps that are zoomable and high resolution, and customizable keys. We've got a lot of other types of visualizations that you can produce just with the basic information shown in the top left, like age category, city, province, exposure type. You can see that we've got a wide variety of charts here. The alluvial flow kind of shows associations from one variable to another. On the right, we've got symptom onset date. And the Gantt chart on the far right here, there's showing a visualization of symptom onset date to the date they were added to, or to the date they were released. And then the date that their test was confirmed is also shown in black. In the bottom left, we show a histogram. So these are the types of visuals that you can produce And I just kind of want to go over some of the design decisions that led to MicrobeTrace. We wanted it to be web-based so that anyone with a modern browser could connect within seconds. No need to download, install, no need to get admin privileges or somebody to help you get it installed on your computer. We wanted it to be quick to deploy. If somebody wants to spin up their own version and set it up behind a firewall, we want you to be able to do that and connect it to literally any file server that you'd like. We want it to be open source, and we've really benefited a lot from getting more public participation, feedback, and scrutiny from public health end users. We want it to be sanely defaulted so that you can create an attractive visualization really quickly, and no configuration is needed in most cases, but that it supports configuration for those like me that want to really create an interesting detailed visual. And finally, we want you to be able to export any of the data that's in here so that you can keep working with it with another program, so that you can submit that to a journal, so that you can blow it up to the size of a poster and go to a conference with it. Those types of things are really important to scientists. So I'm going to switch over to...
I was going to ask a quick question, though, and
yeah.
just getting back to that slide. Can you, and I know you said it, but can you explain again what you mean when you say it's a web-based tool, but we're working with PII, highly sensitive data, especially to be able to do that contact tracing aspect. For the EPIs on the line here, can you explain that what's happening?
Yeah, so thank you. HIV data is probably the most sensitive data that public health typically works with. We have the most rules and laws structured around it. And so we really wanted to build a system that was responsive to those needs. And so this system, even though you're running it on a web browser, and this is where the demo will probably be helpful. I'm in a normal web browser, but if I go to microbetrace.cdc.gov, it takes me to the page. Now, if I were to disconnect from the internet and you still go to this page, once you've already been here once, it will continue to work even though you're off the internet. The key component, the reason that's important is that means that the data is not going anywhere. The data is staying local on your computer. We're very careful when we say, add files, we're not saying upload files, because we don't want to imply that it's leaving your computer. Nothing moves. The second I close the browser, it's all forgotten. You have to reload again. You have to tell us where your files are the next time. So the files here are where they're supposed to be. And so let me give you a demonstration of that. The CDC has very strict rules about where HIV data can live. They have to live on a secured a secured drive that isn't on my computer. It cannot touch my hard drive. Even if it's a CDC computer, that data cannot touch my computer. So we built this program to be able to say add files, go to a mapped drive, load those files, visualize them, but don't move them. And when you want to export data, when you want to move information out of MicrobeTrace back into the secure place that it's supposed to be, you're prompted to specify exactly where that file goes. So does that kind of cover it? I think the whole point here is that we wanted this to function like a program you install on your computer, but to leverage the web browser that's already installed on your computer just to sidestep the whole installation process. And also really gain the security advantages of saying, hey, we're using Google Chrome. Google backs this as a secure feature. The other browser tab that you have open here, if I go to yahoo.com, they're not listening on each other. And we're relying on Google to make sure that there's no crosstalk between the browsers here.
Yeah, I think that's really helpful. So that's to say, we call it a web app, but it's really a browser app. We're using the web to access the application, but in terms of data sharing, there is no data sharing. It's all happening on your local browser.
Exactly. And I'll show, if you want to share your work, we have, made file exports and things for you to do that. but there's no back-end system. There's nothing listening to this. And we really have, like, for example, we're undergoing penetration testing by security experts. We're basically handing them the program and say, break this for us. Tell us where the holes are so that we can be confident in saying this is secure for PII. So there's like a very large contract starting today behind how do we break into this? Is it possible to break into to get the files out? And if they find anything, we'll fix it immediately.
Thank you.
So,
Very reassuring.
Yep. Any other questions before I jump into a demo? All right, so again, feel free to stop me as needed. I'm going to start with the data that I was just showing. It's real COVID data from Korea. So what these look like if you open them up is first what we call a node list. This is a new row for each person. And so this person has a patient ID number here. And they've got information about dates, etc. So this is your standard line listed file. The one that you may not be comfortable with is the transmission network, which is basically patient A had contact with patient B, or in this case, it says this patient was infected by this patient, because that's how they structured their data. I've added fake phone numbers here just to give you an idea of like, this is how contact tracing is probably done right now in the United States. People are going down a list like this, saying person A had contact with person B, with a phone number maybe, and calling them and maybe getting additional information. They're not getting the networked view of who had contact with who in order to kind of see the forest from the trees. So this is what the data looks like. We will drag these two files onto the screen, and the node list needs to be told that it's a node list. I need to tell it where the ID is. You don't have to call it ID. You just need to tell it which column is the ID. So here it's patient ID. We don't have a sequence. If you did, you would just tell it which column had your sequence in it. And then that's it for parameterizing that file. The transmission network is, again, who had contact with who. So we want source or person A to be the infected by, and then target to be the person who's been transmitted to. So person A and person B are connected there, and then we hit launch. Now that's going to render the network where each circle is a person, each line is that connection, and this is a physics simulation. So each little node is kind of pushing against each other node like it's a magnet. The lines are pulling on each other like springs, and then this will lay out the network. So from here, I'll do a few things. First, all the people that are just in the node list and don't have a contact tracing link, we can hide them by saying minimum cluster size is set to 2.
They're the browser full screen.
What's that? Yeah, sorry. And so from here, we can layer on a variety of information. I'll try and quickly reproduce what I had before. So first we could put IDs over top of each individual. We can rescale that size or change the ID's orientation. We can map the shape here to say sex. We can map the size to a variable. In this case, we'll say the number of neighbors. You can rescale that to get an idea of like who's the more centrally connected individual. And then colors, we can map, again, we'll go with gender, and then color the links, the lines that connect people according to the type of connection here. And so you can see that's all very quick to parameterize. And then you can change things. So if I want males to be blue, I can rapidly change that mapping. I can change any one of these mappings. we actually have independent transparency sliders for each type of link. So let's find a common one. We can drag the transparency settings down and like hide a particular type of link in the network. So there's really a lot of robust visual connections or visual manipulations that you can do to the network. But where it gets really useful is having that information alongside something else. So when I select an individual in the network, it'll pop up here on the table as well. And I can bring in some of their information. So like symptom onset date, bring in particular columns. I can select multiple individuals in the table, and you can see those get circled with an orange line that select each individual as a group. We can do things like pin all the nodes on screen if it were working properly. So when you pin all the nodes on screen with this little thumbtack button, you can kind of move things around and say, well, I want these grouped guys that we've selected to be drugged to the side so that you can look at them a little more closely. Again, all of these visuals that we have can be kind of moved alongside one another. And so one of the nice ones is having a map. Actually, let's put the map on the left, the network on the right. And then we need to tell it our mapping information. For us, that's usually zip code or county and state. In this case, because it's a Korean data set, we have latitude and longitude, and it puts it on the map and renders the network over top of it. It does a collapsing thing, which you can turn on and off to give you an idea of density at a particular location. You can also use transparency sliders to get an idea of density at a location. Now notice the map quality is terrible, right? It's very low quality. That's what makes it work offline. If you are on the internet, you can go to the online menu and turn on the base layer or a satellite map, right? And this gives you kind of that infinitely scrollable view that you're kind of used to working with. But keep in mind that this is now reaching out to the internet and saying, you know, request this geographic information, and that's potentially a security issue. And so we have that off by default, again, to prioritize security so that this works in a completely offline environment. So we can put these alongside one another. I had also an age histogram, so we'll drag the histogram down here and parameterize it. And so the variable we're looking at is node age. Notice the color by that applies to the network and the map still applies to the histogram. And so you can even get this differentiation between genders in the histogram. And one of the things that stood out among the early cases of COVID-19 in South Korea is that there was a large number of middle-aged females. And that stands out here. And the reason for that was that was where early transmissions kind of began in church settings. And that's a demographic that most frequently attends church there. And so that kind of comes through in this large data set. So this gives you like a quick view of how you can work with those types of data. We're also generating another data set for that is fake COVID data. To simulate, like if you have one disease investigation specialist going out and collecting contact data, and you want to integrate that with another DIS's data set, along with your typical line listed information about the individual and what their risk factors and symptoms were. So we're still in the process of making this, but I'll give you an example of what that looks like. So here's like 3 different kind of examples of an interview pattern. And you see where there's overlap between the 2 interviews. So this report of contact between 276 and 269 appears in both data sets. And so it's colored with both colors. Down here, if we pin these nodes and pull them apart, you can see that that one appears among these 2 data sets. And so it's colored with both lines there. So we're working with kind of walking through folks of like how you would visualize these data as kind of a PowerPoint document to bootstrap people using MicrobeTrace for COVID-19 contact tracing data. Any questions up to now?
Hi, yeah. So it looked like in the beginning, the data was kind of directional. It was like this person infected this person, but does it have to be that way, or
it doesn't have to be? And so we do have, if we go to the edges and links, there is under shapes and sizes, we have arrows hidden or shown. So the reason we support that is, first of all, source and target, which are the terminology that we use on the file load here of source and target. Those are just common terms used in the field of graph theory, and so they don't really necessarily mean source and target of disease. And in this case, we wanted to support arrows because tuberculosis folks, when they do investigations, they often pay attention as to who named who as a contact and whether those contacts were reciprocally named. They also make estimations as to which infection occurred before another, and can allow them to kind of get a chain of events. So we wanted to support arrows, but not require arrows, if that makes sense.
Yeah, thank you.
Any other questions? All right, so I'm going to jump back to slides real quick and talk a little bit about, let's see here, what we've noticed. MicrobeTrace traffic, we do, when you navigate to MicrobeTrace's or .cdc.gov, it will send you the package of the program, and it will send Google Analytics a note saying this package went here. And so we get an idea of how often people are connecting. And you can see that we have relatively low usage. We have anywhere between 5 to 20 users on any given weekday. And when we have big spikes, they're all on days that we have training events or sessions like today, right? So they're very conspicuous, they stand out, and we notice them. However, beginning in February, we started to have really regular spikes that we had nothing to do with, that were not training things that we held. We still don't really know what they are. Some of them were in foreign countries like Vietnam, where the whole country lights up at the same time, which to us kind of looks like it might be a training session, right? Like somebody in public health may have gotten public health folks to all connect and do a training session all at the same time. And so we're very hopeful there. Our monthly users has continued to increase since our launch about 2 years ago. And our daily users looks pretty similar. You can see there's a huge difference between weekday and weekend, and that's just because most of our people are in public health and they don't work weekends. But as I showed you just a moment ago, we're working on a training data set that has characteristics that are common with COVID with respect to symptoms, comorbidities, that looks like contact tracing data, and that is kind of a hold your hand walk through how to make these types of visualizations. So that's in the works, and other pathogens that have been evaluated with it on this list, and this is growing pretty rapidly. So there's really a broad use case, and I think we'll put this last slide up for folks in terms of resources. The main tool is up there at the top. Again, you will need Google Chrome to access it, or a modern browser like Edge or Firefox. Internet Explorer won't work. Unfortunately, our Google Analytics says that about 8 to 12% of our traffic still goes to IE. So someday that will end. We have a flyer on our GitHub page. You can Google the MicrobeTrace flyer. That'll give you kind of a quick rundown. There's a very detailed manual that walks through every visualization. There's also a YouTube webinar that's about an hour long that's cut up into different sections of my ugly mug going through everything that I just did here. And then there's our GitHub page where you can find all of these things above, as well as the sample files that I'm describing, Wikipedia entries if you're interested in deploying your own. And again, one of the things that we're planning to develop here that we've recently kind of been given the green light to do is develop a Docker container or a menu of Docker containers that would allow you to connect MicrobeTrace to any arbitrary resource. And our first ones that we'll target will be like Access databases, RedCap instances, SQL, basic query structures that will make MicrobeTrace kind of interoperable with other systems. So with that, we've got lots of time for questions and discussion.
Thank you. Live demo. Els, I had a question. So you definitely showed the utility in terms of contact tracing, especially when you have that epi data and even all the different colors there and the different visualization options, where hopefully the epis on the call can already see the utility in that case. Now, in addition to that, if we wanted to add the genomic analysis aspect to this, is that as simple as dragging a NUIC file onto that same screen at the same time?
As long as your IDs match, that's as simple as dragging a NUIC file on. So I'll start over again. So if you've got your sequence file, that's one way to drag it on, and you would just say it's a FASTA file. If it's a NUIC file and you've already done the bioinformatic work, then just tell us it's a NUIC file and we'll be done with it. So
well, we can put assemblies
there
too. Sorry, we could do assemblies and it'll do the alignment and everything's there as well.
So because it's in the browser, we're really limited by computationally.
Yes.
So we would prefer that, like, if it's a FASTA file, narrow that down. If it's a TB, for example, narrows it down to just their SNPs. Cyclospora has a huge array of genes that are recombinating because it's a eukaryotic organism that sexually reproduces. They do their own modeling and give us a distance matrix and just click matrix here, right? Okay. So it depends on like whichever format you want. We natively take things out of bionumerics. So like if you're getting a distance matrix, whether it's the half matrix, like the top corner, or the full matrix, we don't care. We'll take it as a pairwise distance list. So there's like a bunch of different ways to get the data in. If it's a tree, we'll view the tree in the NUIC structure. But we will also measure the tip-to-tip distance. So let me go to our GitHub page, and I'll show you what that means with some sequences. So here under demo, we've got a NUIC file that I can get the raw file, notepad.
And just so you're aware, Els, you're getting a couple questions on the chat there too.
Okay, great. Thank you. So let's say phylotree.nwk. So if I go back over here and drag this NUIC file, tell it it's a NUIC, and launch, it is now looked at the tree, measured the tip-to-tip distance between those trees, and constructed a genetic distance network. Now you can decide what threshold you want. Our program defaults to 1.5%. That may not make sense for you. Here, it may make sense to kind of capture this first peak in this histogram, as opposed to this large peak way out here where everyone's connected to everyone. But the point is you can kind of dynamically pick the threshold that makes sense for your bug. And so if you want to just continue to look at the tree, we've got a phylogenetic tree viewer as well that is relatively robust. You can do a lot of the same things like add your labels. Let's see, labels, ID. You can resize those, map different shapes to different things. So we have a lot of that basic functionality. Let's pull up the chat. Saying they defaulted is useful, thanks. So I'll start with a question about hepatitis data. It's actually really interesting. If you are aware of GHOST, the Global Hepatitis Outbreak Surveillance Technology, the latest version of GHOST has a button there where you can launch their data that they compute in MicrobeTrace. So they actually serve up MicrobeTrace as a part of the GHOST platform. So basically, they've just said, this is a more robust visualization tool. It's going to end up in a lot of the same capabilities, or doing the same capabilities that we are. And so let's just combine forces, and they serve MicrobeTrace as part of their visualizations. So I wish I had a slide of that. I don't have one handy. Description, website to explain the tool for EPIs. So yeah, that's a good question. I think our manual via email would be great. Okay. So if we've got, we've basically produced a flyer exactly kind of for that situation that we could provide and make available. So we've got kind of a slick sheet of what, why, how, where, who, with all the information, some of the visuals, some of the input types of files. So I think I'll just link up with Kevin and other organizers to make sure that those documents are available to everybody from the chat. And then I think I wanted to try and talk briefly about kind of how we could use this for COVID at a larger scale and comparing it to NextStrain. So let me switch over to NextStrain. So this NextStrain is a wonderful tool. I don't want to disparage NextStrain in the slightest. And so don't take these comments that way. Given that this is a global outbreak, given that this is as big as it is, I don't find this visual even remotely helpful. There's just far too much information on here, even in terms of it being interactive. You know, it becomes useful once you've narrowed it down to a very small tree, right? And that's what it was really used for in the beginning. That's what it was developed for, data of not quite at this scale. I think networks are much more adept at visualizing these types of data, and we are developing MicrobeTrace to kind of be able to do that. The scale of problem is obviously difficult for us. You know, what you see here is 2,700 genomes. They even have trouble getting to the tens of thousands of genomes that are available on GIS, AID. But ultimately, we would like, and this is a program called Gephi, to be able to visualize the COVID network in this context. So what we have shown on screen is a circle represents a unique SARS-CoV-2 sequence, and a line represents kind of the closest genetic neighbor. Where there are cycles or kind of equidistant options, we draw both lines. We don't make a decision of picking one or the other if both are equally applicable. But this, to me, makes, even though it is relatively jumbled, it makes a lot more sense. I've colored, as you can see, by region, and you can automatically see that there's kind of two major introduction events into the United States or into North America colored in yellow. And I think that jumps out at you much more than if we were to, I guess I can reload that, try and garner that from this type of a visual. And so our interest is kind of, how do we take what can be done in a network realm, make it web-based, and make it functional for public health? And we're moving in that direction. We're not quite there yet. If you look at our development version of MicrobeTrace, the non-production environment, there's this pixie view that we're working on. This allows us to generate much larger networks much faster. And so I'll show you kind of on our issue tracker on our GitHub page what that looks like. This network is a very large network with many, many nodes and things on screen. MicrobeTrace could not handle visualizing that very smoothly. And so we're working on kind of developing the technologies to be able to visualize large networks and really give kind of context. And again, the ability to really customize the visual here. So if you wanted to take your own implementation of NextStrain, you can do that and set it up on your own environment. I talked with Kevin earlier today, and it sounds like there are a number of states that have set up kind of their own local version of NextStrain. We have one at CDC as well, and we use it for various analyses. The problem that I run into is if I want to change colors of something, I have to actually edit the underlying files. And it's not just say like red to green. I have to go look up the hexadecimal code for red and green to figure out how to change those. I have to manage like multiple files to be able to do the things that I can rapidly do with a few drop downs and slider bars in MicrobeTrace. So like we don't have the robustness and the bioinformatic capabilities, I think, that underlie NextStrain that can do large genomes and those types of things because they're running on a big server that can handle large volumes. And so there's some pluses and drawbacks for both. Ultimately, if you want to make a very customized, informative visual that layers on a bunch of information that can help you understand your local problem and really see the forest from the trees, I think MicrobeTrace is a much better tool to do that. And it also, when you do start to get the contact tracing data, you can then begin to layer it on and they directly overlap with one another. You're going to have a hard time doing that with something like NextStrain. Like how does this link between person A and person B relate to this series of branches that might connect person A's virus and person B's virus?
Yeah, Els, I think that's definitely like the point of the handshake where we're kind of the challenge is because we can generate those bioinformatics analysis, we can get the assemblies, we can get those Newick trees, but then adding that layer of the epidemiological data to overlay on it is a translational step we're trying to figure out. So this seems like a solid solution that public health labs could use with their health departments, where we could share just a Newick file with our epidemiologists, and then through this platform, they can also go in there and add layers of the contact tracing information to further refine their outbreak investigation.
Right. I think the benefit here is really the rapid iteration that you can do on these types of figures. In the past, I used to attend meetings. I would show up with a bunch of figures, and questions would come up, and I would have to come back the next week with new figures that address those questions. With this type of a tool, you can do it on the fly and actually like address those types of questions in real time and really iterate over a bunch of different figures and see how they interact. So it just, there's nothing new here. These are all things that you can do with other tools that have been out on the market. Like I showed Gephi, I've been using Gephi for years. It just kind of is a force multiplier because it puts them all in one place.
Yeah. That's really powerful. And I'm curious to know if there's any EPIs on the call. I know a couple of Virginia folks I see on the call here. If they can comment in terms of how applicable this system might be. I think for me as a laboratorian, this seems like it's really consolidating a lot of our efforts into a single platform in terms of wanting to combine the genetic analysis with the contact tracing information, all the other epidemiological factors. But how feasible does the integration look for some of the epidemiologist folks on this call?
Logan, it looked like you're about to say something before that.
Yeah. So I guess that is when my question actually kind of ties in a little bit with what Kevin kind of led in, and seeing as none of the EPIs are brave enough to answer, I guess I'll kind of go ahead. I'm just kind of wondering, with this type of network, it seems like it is really dependent on kind of like good epi metadata. And I don't know, like for us in the lab, we're generating more laboratory specific data. And so I'm wondering, have you guys done any sort of validations of like,
Yeah,
using just like the laboratory kind of data and seeing how
right.
well it aligns
So
with the epi data? And
I think it's going to depend on every situation. I'm working on one right now where we're looking at epi data of monkeys and whether they had contact with one another and different types of contact and integrating that with different genetic networks. And so like this is just a really great tool. I've got 3 different genetic networks here that I can drag on and then layer on the individual level information about those and see where they kind of agree and disagree. I forgot to set a distance parameter, but ultimately, I guess the point is you can evaluate different methods, different approaches against one another in isolation of your other information. So, I would say if you want to look at what the genetic signal looks like for this region and compare that to another region, you can readily do that and see like where 2 different methods might support one another, where like this link between these 2 cases is only supported by this one method. This link between these 2 is supported by 2, and then this one looks like it's got 3 different colors associated with it. So there's a lot of different use cases, but you're right, when it comes to like how well does it correlate with, needle sharing or sexual contact, I think, that's a hairier issue that I'll let the EPIs tease out for me.
thank you. Yeah, that's really helpful.
And there are some comments populating the chat there. Catherine, thank you for showing some perspective there for the Virginia EPI side, noting that this could be something helpful, especially for things like COVID, where a lot of information is known about these patients, but for things where there are fewer cases, it may not be as helpful for E. coli. And I think that's true. I think for those enterics, perhaps staying with the PulseNet tools that have been built specifically for those kinds of investigations, that may be the more appropriate approach. But for something like COVID-19, hopefully this is a tool that we can start adopting here, even in Virginia.
Yeah, there's a good point raised by Taj. He says, but in general, this is largely descriptive and relies on some knowledge of what is meaningful relatedness among epi and genomic data. And that's basically what Logan was pointing out as well. I think ultimately, there is still some implementation science that needs to be done in every single silo that we focus on for these bugs, because the answer is going to be different for every type of transmission. There's work being done in Europe. Christoph Fraser is kind of leading the effort to kind of come up with confidence intervals for directionality. And a lot of that work, and they're doing some NGS work instead of just looking at these short sequences that are conveniently available for public health. So there's definitely a lot of science that needs to be done to figure out exactly what the utility here is for us. And I realize we're definitely kind of on the cutting edge, but it seems to be a much more useful tool than trees were in isolation because of how readily it integrates with the contact tracing data. Now, whether or not that's valuable or accurate contact tracing data, I think we need to do that integration first to vet that question.
And kind of, I guess, going off of that too, then, it seemed like you guys were doing some kind of machine learning algorithms applied to the data to tease out like different variables that might be indicative of like transmission and what things like that could affect the data. So I'm wondering, are those available through here too, or is that something that would be separate?
That's not through MicrobeTrace. I mean, those are definitely things that we're doing kind of in our own work. But yeah, I mean, we'd be happy to interact with you over that.
Yeah, I think that would be a really interesting area of study for public health between kind of the laboratory and
Ultimately, we need the contact tracing data to overlap with it to do that type of validation work. I think that's really what needs to happen. And the biggest problem that I see right now is that when there's an outbreak, they often stop contact tracing when they find a negative. So you're unable to like see what differentiates the contact network of a negative individual from a positive because you bias it in the data collection effort to begin with. So I think there's still a lot of work to figure out like how do we actually translate this into real public health and solid science. But you know, this is definitely a fruitful trajectory.
So one comment I want to add on in terms of, you know, we actually have to heavily rely on the EPI's contact tracing rather than this, is we have been working on our EPIs to look into some of the long-term healthcare facilities. And you know, we're basically sequencing as much as we can within one facility. And the trend this project gearing into is they try to use the genetic readiness data, the phylogeny data, to inform them who give COVID to who, which is a little bit of overreaching as far as I think. Basically, they were asking, oh, okay, there's 2 nodes in these branches, but they're in 2 separate floors, and how could they link? And I have to like repeatedly remind them, one, we have not sequenced every single one patients. And what phylogeny shows you is not necessary to represent who give COVID-19 to who. We still need heavily rely on the EPI's contact tracing and interviews to build up this bridge. And also be mindful that, you know, this is a new virus that gets transmitted in an extremely short period of time. And the mutations, the accumulated mutations is going to be minimum, and how this evolutionary process can inform the transmission event is still need to be, you know, there need to be more research on that point.
Yeah, I don't disagree with that at all. I think folks at CDC and all over the world are kind of investigating that point. Ultimately, you know, it is a very slow-moving virus by comparison, and so there's a lot of similarity between sequences and individuals, and there just may not be enough resolution to say, you know, transmission is confirmed or not, and we may just never get there. I think that answer is going to be different depending on the bug, its rate of evolution, the length of the sequence we're looking at. There's a lot of questions that feed into that. And while, you know, I think ultimately right now, we're not even at the point of looking at contact tracing networks. Like I haven't seen any yet, other than what came out of Korea. So I think we're still building up the basic foundations of how do we measure this? How do we look at it?
All right, thank you, Els. We're at the top of the hour here. So wanted to thank you for giving this presentation. Thank you everyone for all the really strong questions and perspectives. As was discussed in the chat, I'll be able to connect with Els and his team to make sure I distribute all of those manuals and flyers for MicrobeTrace to everyone in the StaPH-B network. Other than that, we'll make these slides available on our website and also the recording. And also this is, I think this is probably my last StaPH-B call, so at least leading it. So thank you guys for the number of years allowing me to be a part of this thing. I really appreciate everything. So have a great weekend, everybody. I'll see you guys next time. Thank you, Els.
Thanks,
Thank you so much.
Thank you.